Okay, today I'm going to tell you about some work that we've been doing on trying to understand the role of iRhom2. So iRhom2 is associated with palmoplantar keratoderma, but when we identified the mutations, very little was known about this protein. So that's why I'm going to say we're trying to understand the biology from a standing start, into the unknown. So story all started by studying an autosomal dominant palmoplantar keratoderma syndrome called tylosis with esophageal cancer. So the patients present with a focal palmoplantar keratoderma on the soles of their feet and on their hands. And you can see how thick the palm epidermis is from these top patients compared to control. Now, as well as presenting with a skin disease, these individuals also have a high risk of developing esophageal squamous cell carcinoma. So after quite a long journey, we finally identified the cause of this condition, and it was due to missense mutations in a gene called RHBDF2, which encodes the protein called iRhom2. So what does iRhom2 do? Well, it's a 7-transmembrane protein. It has a long cytoplasmic amino terminus, which is where all the missense mutations cluster in a highly conserved domain. As I say, it's a member of the rhomboid protease family, and some of these are serine proteases. However, iRhom2 does not have any protease activity. It has lost its protease activity, and so that's why it's called an inactive rhomboid protease. So we really didn't know much about what iRhom2 would be doing in the skin and the esophagus. We knew it's called rhomboid because basically when you knock it out in Drosophila, the larvae have a rhomboid shape. And they did some studies been done to suggest there's a link with rhomboids with EGF signaling, growth factor signaling. So what we did was we dug out the keratinocyte cell lines that we made many years ago from these patients and hadn't done anything with them until we identified the disease gene. And the top keratinocytes, which are the patient keratinocytes, were compared to control keratinocytes. And we're just measuring proliferation. And in medium with serum growth factors added, both cell lines grew very similar, no real difference at all. The real difference happened when we then culture the cells in medium without serum and growth factors, without serum and added growth factors. As expected, the control keratinocytes stopped proliferating and just sit around not doing very much, whereas the top keratinocytes kept on growing, they kept on proliferating. Another way of looking at this aberrant growth factor phenotype is by doing a monolayer scratch assay. So this is like doing a wound healing assay in monolayers. And you have your cells in the culture dish, and then you make an artificial wound with a pen or something like that. And then what you do is you visualize the cells over time to see if the keratinocytes can migrate and proliferate and close the wound. So this is done in media without growth factors added, and you can see the control cells, nothing happens. Whereas the top keratinocytes very quickly start to fill the gap, and there's complete closure after 2 days. So what is causing this change in cell behavior? So one of the highly expressed growth factors in keratinocytes is a protein called Amphiregulin. And Amphiregulin can be secreted from keratinocytes. So what we did was we did the same scratch assay in control and top keratinocytes, but at different time points, we took some media and measured to see how much Amphiregulin had been secreted. And you can see in the top keratinocytes, there's bucket loads of Amphiregulin being secreted compared to that of control. So basically what's happening is the top keratinocytes, which have the iRhom2 mutations, are adding their own growth factor to the media. And indeed, if you took conditioned media from the top keratinocytes and gave them to the control cells, they would start to proliferate in a similar fashion to top keratinocytes. So this excessive growth factor shedding, how is this happening? Now, at the same time as identifying the iRhom2 mutations, we also had identified by studying a recessive condition of an inflammatory skin and bowel disease, are patients who had loss of function mutations, homozygous loss of function mutations in the gene encoding the protein called ADAM17. ADAM17 is also known as TACE, or TNF-alpha converting enzyme, and it's a membrane-bound sheddase which is involved in the regulation of a number of ligands. This is a cartoon. This is ADAM17. So when this inactive propeptide domain of ADAM17 is removed at the cell surface, it can then, like a scissor action, clip away at various ligands. So it's cleaving these ligands and releasing them into outside the cell. And it has a number of sort of substrates, ADAM17, and including members of the growth factor family. In fact, it's very specific for the shedding of Amphiregulin, as well as other cytokines and receptors. So maybe iRhom2 in some way was linked in with ADAM17 because of the excessive Amphiregulin secretion. So one of the things we did was we did some immunohistochemistry where we stained iRhom2, and you can see in the patients who have no ADAM17, there is a dramatic reduction in the expression of iRhom2 compared to control, suggesting there is a relationship between iRhom2 and ADAM17. To develop this further, we then immunoprecipitated iRhom2 in keratinocytes, and then did a Western blot with ADAM17. And you can see that ADAM17 is in this complex of iRhom2, suggesting that it may interact directly or very closely with iRhom2. So the other thing we did was we did 3D cultures and just looked to see where ADAM17 was expressed. And you can clearly see in top keratinocytes, there is lots of ADAM17 expression and lots of it at the cell surface. So what we think is happening is that iRhom2, well, it's been proven now that this is what's happening, iRhom2 interacts directly with the inactive form of ADAM17 in the endoplasmic reticulum. This complex then translocates to the Golgi, where another protease called furin removes this inactive propeptide domain, and then iRhom2 and ADAM17 then go to the cell surface where ADAM17 is active, and it can cleave all these ligands and release them. So basically the mutations, the top mutations in iRhom2 are gain of function in nature and are increasing the sheddase activity of ADAM17. To show it's ADAM17 dependent, we then went back to the ELISA assay and we looked at the shedding of Amphiregulin and another growth factor, TGF-alpha. So in grey is comparing top keratinocytes with control. You can see how much shedding there are of these growth factors compared to control. And then when you inhibit ADAM17, this is a small molecule inhibitor, you can see this shedding is dramatically reduced. So this excessive shedding that we saw originally in cell culture Amphiregulin is dependent on ADAM17 sheddase activity. So that's one function for iRhom2. The other thing we thought we'd do was we looked at a mouse model that had been made, and this is a knockout of the gene RHBDF2, which encodes iRhom2. So the mice look phenotypically normal. There actually seems nothing wrong with them at all within the lab environment. However, when you turn them over and you look at their foot pads, they were white and translucent. Fully intact, there was no blistering or anything, but they were very different to that of wild-type mice. And then when you do histology, you can see that the pores are dramatically thinner from the knockout mice compared to controls. And it was very specific to the pore skin with very little difference, if any, in the back skin. We then used CRISPR-Cas9 technology, and we knocked in an equivalent TOC gain of function mutation into the mouse germline. And what we saw was the opposite of what we saw with the knockout. We saw the pore skin of these mice was dramatically thicker than that of control. So iRhom2 is somehow regulating the epidermal thickness of the palmar plantar. Because iRhom2 interacts directly with ADAM17, we postulated that maybe iRhom2 may interact with other proteins. And so what we did was we performed a yeast-2 hybrid screen where we used the amino terminus of iRhom2 as a bait. So this is the region of the protein where the missense mutations are found. And we screened this bait in a skin library. And we identified one of the positive hits as being the wound healing keratin, keratin 16. So how can we confirm this interaction? Well, we did a number of experiments. One is we overexpressed iRhom2 with a GFP tag in keratinocytes, and then did immunocytochemistry to see where the expression of keratin 16 was. And where you see this orangey yellow, the pseudo color here, you can see that there is definitely a co-localization of iRhom2 with keratin 16. Another way of visualizing this is to do an immunoprecipitation. So basically, we pull down keratin 16, and then form Western blot with iRhom2 and its close gene member iRhom1, and you can see that iRhom2 but not iRhom1 can interact with keratin 16. And note there is much more interaction in top keratinocytes between these two proteins than in control. Another technique to visualize protein-protein interaction is called the proximity ligation assay. And this measures protein interaction within 40 nanometers. So they're maybe not definitely interacting, but very close to each other. How this method works is that you use your two antibodies, so K16 or iRhom2, and then you have a secondary antibody that then binds to these antibodies. And at the end of these antibodies, there's a DNA tail, there's an oligonucleotide sequence. And when if these two proteins or antibodies are close together, these oligonucleotide sequences ligate to each other. And you get like this, and you add DNA polymerase, and then you get this like rolling circle amplification. If you go in with a complementary oligonucleotide probe to this sequence here, which has a red fluorescent tag on it, you can then visualize where in the cell you get protein-protein interaction. So if you go to the control keratinocytes, first of all, you can see there's red dots, and this means that keratin 16 and iRhom2 are interacting with each other. But what you can also see is there's much more K16 iRhom2 in the patient keratinocytes, the top keratinocytes, compared to control. Now, keratin 16 is a type 2 intermediate filament protein. Sorry, is a type 1 intermediate filament protein, and its normal binding partner is its type 2 keratin, keratin 6. So we did the same experiment looking at K16, K6 interaction. Sure enough, in control, we see lots of positive signals indicating these two keratins are heterodimerizing and forming filaments. Whereas if you look at K16 and K6 in top keratinocytes, you can see very little interaction. So it looks as though iRhom2 is regulating keratins K16 and K6. So we then went in a little bit more detail. And so we just did Western blotting of control and top keratinocytes. And you can see there's much more keratin 16 in top keratinocytes and much more K6 in control keratinocytes. We then did the 3D skin cultures. And I just want to look at if you just look at the top panel here, this is without any genetic modification. You can see there's high levels of K16 expression in TOC. However, if you then use a short hairpin iRhom2 retroviral construct into the cultures, which basically causes RNA interference of iRhom2, you can see without iRhom2, you get a dramatic reduction in the expression of K16. When you look at K6, the opposite is happening. So without any genetic modification to iRhom2, this is the expression of K6. But when you knock down iRhom2, you get a dramatic upregulation of K6. So iRhom2 is regulating the dynamics of these two keratins. So we then went into the mouse model, the knockout mouse, and sure enough, in the poor skin of control mice, you see K16 and K6. But look, in the knockout, very little K16, but much more K6. So this is modeling exactly what we saw in the 3D cultures when we knocked down iRhom2. So iRhom2 seems to be regulating the organization of the cytoskeleton. So what we wanted to do is we wanted to see how these cytoskeleton will respond to cell stress. So what we're doing here is something called cyclical stretching. And so we're stretching the keratinocytes over a short time period. This is 4 hours. So you can, and then we're staining with keratin 6. So you can see when you do control keratinocytes, after 4 hours stretching, you start to see increased expression of K16, but notice it's forming this perinuclear localization. TOC keratinocytes, even without stretching, are showing this perinuclear organization, which is much more pronounced once you do cell stretching. And you can quantify this and show that there's much more perinuclear keratin 16 in TOC keratinocytes compared to controls. So what do we think is happening here? Well, we think when the cellular stress, i.e. in disease or in wound healing, iRhom2 regulates the relationship between K16 and keratin 6 and switches it so keratin 16 is expressed and is associated with iRhom2. And it changes the cytoskeleton so it becomes more perinuclear. And this is a phenotype which is often seen with keratinocytes, which are involved in reepithelialization after wound healing. So as well as the change in the keratin, we also see a dramatic upregulation of ADAM17, so increased growth factor and cytokine production. So this is our working model. What's going on there? Another question we wanted to ask was, well, what is regulating the expression of iRhom2? What causes iRhom2 to be activated in certain disease situations, like wound healing? So one of the most studied transcription factor in the skin is P63. And so we focused on P63, and there was available a ChIP-seq dataset, which is looking at P63 protein interactions with the DNA across the whole genome. And this data set was available, and we would then said, well, is there any evidence that P63 could be regulating iRhom2? So this is the gene RHBDF2, which encodes iRhom2, and you can see there's a P63 peak here in a downstream presumed enhancer region. We then cloned this region and then look to see if indeed P63 could bind to that region, and sure enough it does. So again, just want to reiterate, P63 is a key transcription factor in the skin. It seems to be involved in the development of the epidermis, but it's also expressed in many tumours, and of relevance to TOC is also associated with esophageal cancer. To follow this up, we then knocked down P63 at a protein level in keratinocytes, and you can see there's a dramatic reduction in iRhom2 protein expression. We wanted to look at the messenger RNA level, of course, and so when you knock down P63, again, you see a dramatic reduction in the gene expression of iRhom2, RHBDF2. So P63 is regulating iRhom2 in keratinocytes. We then went back to the knockout mouse model to try and understand a bit more about how P63 was regulating iRhom2. And so I'm just focusing here on the back skin of the control mouse versus knockout. And you can see in the back skin of knockout mice, there is much more P63 protein expression compared to control. You then go to the paw skin. So you can see in the wild type mouse, there is lots of P63 expression, whereas in the knockout mouse, there is very little. So there seems to be this context-dependent signaling expression going on here. And so the working model we have is that in a normal homeostasis, P63 regulates iRhom2, which in then represses P63. Whereas in disease situations, such like TOC or in wound healing, you see p63 activates iRhom2, who then feeds back, and you get high levels of iRhom2 activity. And this then affects proteins such as ADAM17, but also we've shown it affects downstream pathways, which are involved with regulating apoptosis and also oxidative stress. So what I've shown you here is that from a standing start where we knew very little about iRhom2, we find that iRhom2 is actually regulating many key functions within the skin in terms of regulating the shadows, ADAM17 and cytokine growth factor signaling. Also seems to regulate the cytoskeletal stress response. And we have this new signaling access, p63 iRhom2 access, which can regulate stress-associated pathways, but it does it in a context-dependent manner. And this axis also regulates cell survival and oxidative stress. This cartoon puts together some of the things I've talked about today, but also some of the other things that people have found regarding to iRhom2 function. And so you can see that there's actually very diverse roles. So as well as the ADAM17 regulation and the signaling through p63 and the keratins, it can also regulate the STING complex, which is involved in the innate immunity response to viral infection. So iRhom2 appears to be a very fundamental molecule in the keratinocyte and also in the immune response. From that, I will take some questions. Thank you.